Okay, welcome back from this very small break, I admit, to this session on animal welfare in wildlife research and management, the 3 R's in animal wildlife. My name is Tom Bengtsen. I'm a veterinarian from the Danish Veterinary and Food Administration. They are working with both laboratory animals, Danish 3 R Centre and the Danish Centre for Animal Welfare. So, of course, a session combining 3 R's and animal welfare is just what I would like to hear. And we'll start off with Johan Lindsjö from the Swedish Centre for Animal Welfare, and he will talk about 3 R's in wildlife research and management. You're welcome.
Thank you very much for inviting me to this very interesting seminar. I will talk about the 3 R's in wildlife research and management. And this is the outline. I will first walk you through the principles and applications of the 3 R's, then go into the 3 R's in wildlife research, dip into legislation and guidelines, and then finalize with the 3 R's in wildlife management and how it can actually overlap with research sometimes. So these two pioneers, Russell and Birch, they wrote the principles of humane experimental technique, where they defined the 3 R's for the first time. And those are, as most of you probably already know, replace, reduce, and refine. And when you replace something, you use an alternative to animals when you do your research. And you reduce, you can use fewer animals to get the same result, or you can use the same number of animals, but then get more information for those. And finally, refinement is to alleviate pain, distress, and suffering, but also to enhance the well-being of these animals used. Importantly, this should be done and at the same time keeping a good level of the research quality. And the concept of the 3 R's was originally developed for improving laboratory animal situations. So the replacement was, for instance, in vitro methods in cell cultures and silico, computer modeling, predicting different scenarios. Reduction is to optimize the study design through, for instance, through statistical improvements. And refine is about care, handling, and housing of the animals that you then use. And then into wildlife research, which I've chosen to define for the sake of this presentation, studies on free-ranging wild animals. And it can cover a variety of areas, as you can see: biology, ecology, abundance, behavior, and health, disease transmission. The animals can also be indicators of environmental health, and lately it's actually used for studying human disease as well. So then the question is, are the 3 R's applicable on wildlife research? Well, we'll start to look into some challenges and limitations when you look at the 3 R concept, and you see that there are a few differences between laboratory and wildlife research when it comes to species, research environment, purpose, and design. And first of all, when you do research on traditional laboratory animals, they also often are models for human physiology and pathology processes, while the wild animals are often subject for the investigation themselves. In traditional laboratory animal research, you often do experimental manipulations, and in wildlife, at least until now, it's often descriptive research. And of course, there is a big difference how standardized the research is when it comes to environment and animal, which are sometimes inbred when it comes to laboratory animals. There is acclimatization of the animals, there is standardized methods. And when you look at the wildlife then, as you understand, the environment can be very different species, different populations, and also different genetic background. Another important point is that animal welfare implications and indicators are largely unknown in wild animals still. And finally, there's still a lack of awareness and knowledge and education among management and organizations, researchers, staff, animal welfare bodies, ethics committees, and authorities. So back to the question. Are the 3 R's applicable in wildlife research? Well, if I shouldn't finish my talk after 4 minutes, I have to answer yes, of course. And I'll go through some strategies, 3 R strategies. So we start with the replacement. And this is a long list, so please read. For instance, you can use non-invasive alternatives to collect hair, feces, and urine, for example. You can also collect environmental samples, such as DNA can be extracted from soil and water. And you can also do observations directly and through cameras and video films, for instance. You can predict abundance, behavior and movement and disease transmission through computer modeling that we call in silico. You can use archived tissue samples. You can analyze data that's already been collected. And you can use models with immature animals from vertebrates and invertebrates. And then there is debatable point here, if you can use species that are less important than important keystone species that fulfill a very important role in the environment. Is this really a replacement? Well, not per definition, according to the EU directive on the protection of animals used for scientific purposes. So, when you reduce animal use, you can use appropriate and efficient experimental design, as I mentioned before, including statistics. So you can, for instance, try to minimize the sample size, the number of animals, and try to have enough power, still maintain the power of the analysis. And you can utilize species, gender, and age specific experimental design. So, for instance, if you trap an animal, you can use a certain bait. And you can also use time and location when you capture animals in your research to actually target specific gender, age, and species. And of course, maximize the use of the sample you actually get. Trying to minimize capture of non-target animals. Try to coordinate between research groups and fields, whatever results you want to get. So you have one group of animals, but you can use different projects and get samples from the same group. And of course, you can use biobanks as well. Share and publish data is important, and you should also, don't forget that you should also publish negative results, so people know when you fail, when things do not work. Try to avoid, if possible, repeating captures and, of course, studies. And then you can combine replacement with in vivo, and that will then be a kind of reduction. So for instance, if you get hair from hair snags from barbed wire traps from an animal, that you combine with whatever you get from animals that you actually capture. And by then you can actually reduce the number of animals you really need for instance, an hormonal analysis in hair. So finally, refinement. Less invasive collection of biological samples, of course. And then use the best practices for capture and handling. Cause as little stress and harm as possible. Training of field persons is very important, so you know what you do, so you don't cause unnecessary suffering. Collaborate with manufacturers to get the best equipment. Also, don't forget to define humane endpoints. You do that, you have to do that according to your ethical approval, but you have to think about when is it actually best for a wild animal to do that. Can it survive if you release it, for instance? And then, of course, appropriate euthanasia methods for both the target species and the non-target species. And a very important point here, that is often important. Sometimes you forget that, but you shouldn't disturb the environment for these animals. Den sites and nests, feeding areas, and social structures. So just an example from the east coast of Canada, Grand Manan, where there is a whaler research station who collaborate with fishermen. Porpoises, the small whale species, we also have around Scandinavia here, are accidentally by-caught, and they are then saved by the fishermen together with the research groups, and they take the chance to do research on these animals. So by using those animals instead of capturing other animals, you reduce the number of animals used. They're using very fine methods. This young, nice, handsome gentleman here is using a sponge to moisten the animal. The vital signs are always monitored, and then you get them out as soon as possible and release them after samples are taken and there are tags are put on them. And this tissue bank can then be used for other experiments, and that's part of the replacement. Similar group have been looking into shearwaters, how they migrate, and they are then very carefully, in this project, they really put lots of effort into how many animals do we really need, how big sample size do we have to get the results we need. And then they capture them at night, swooping them up with a net, put them in a bag so they are more calm and potentially less stressed. They use very fine methods to sample them, and then the best, at that time, the least invasive way to put a transmitter on them. And then they're released, and that's part of the refinement. So quickly then, when in the research process are we using the 3 R's? Just read through quickly while I'll moisten my throat here. So that's the full research process we have here. Start a hypothesis, you apply for the research funding, you actually using the animals. If you come to that conclusion, you analyze and you write up, and then you publish and you present your results. And if you look through the literature, it's very often that you see it's about refinement. That's where you put lots of efforts into research in general. However, it's actually legally required and also best for the research to use it straight through the whole process and start early, because then that will then influence the research process straight through. So 3R thinking is important straight through the process. A little bit about the legislation and guidelines here. We have in EU, we had the directive on the protection of animals that were implemented in the Swedish legislation 2013, and likewise should be implemented in other EU member states. And it says, or include animals that are used for scientific education purposes, that are subjected to pain, etc., that are higher than that caused by the insertion of a needle. In the Swedish legislations, welfare legislation, we kept our previous definition that was the purpose to define if it was research or not the suffering itself. So, however, it does not include practices undertaken for the primary purpose of identification. And very importantly, all research procedures should be in compliance with the 3 R's. Wild animals should not be used unless the competent authority has granted an exception. An endangered species, of course, a minimum should be used. Importantly, training education is emphasized, and then the setup of animal welfare bodies for breeders, suppliers, and users of animals to actually give advice on the animal welfare and the 3 R's for these people handling these animals. And I've chosen the 3 websites which are set up by platforms to 3R centers around the world that I find most useful when it comes to looking into best practices and guidelines for wildlife research. The first one, Norway's Norecopa. Then you have the British one, and then the NC3Rs, and then finally the Canadian Council of Animal Care and Science. And they're very, very helpful if you want to look into that, the 3Rs for wildlife research. So, what about 3Rs in wildlife management? Now it's getting a little bit complicated here. I have decided, not only me, it's quite a common definition, that when it comes to management, you're still looking to gaining some information and knowledge, but you're assessing and monitoring different things: population size and movement, diseases, environmental health within conservation, and even invasive species. They are sometimes based on legal requirements, national and international ones, or decisions from authorities. And they can sometimes overlap. So for instance, if you capture an Easter size and then sample an animal, get that information, it can sometimes be counted as research, given the purpose, or as wildlife management. However, research and management differ in legislation in many countries. For instance, in Sweden, where research is within the covered by the Animal Welfare Act and its regulations, where it's quite detailed how you should protect these animals and how these should be controlled by official animal welfare controls, go through ethical review and approval, and then, of course, requirements of the 3 Rs. While in management today, covered by the hunting, fishing, and some environmental acts are not, are either completely lacking these requirements for animal welfare, or they are very vague. So, in summary, research, 3R, management, no 3R. So animals are then exposed to can be exposed to similar activities and methods, such as capture, immobilization, marking, sampling, transport, euthanasia, etc. And as you know, this can all cause stress, fear, and pain in these wild animals. But they have different protection. Including the requirements of the 3 Rs. It has been argued, and it's still argued, that wild animals that are kept by humans, although short-term, and are exposed to invasive procedures, that all these should fall under animal welfare legislation. In addition, through an ethical review and approval, you can also make a cost-benefit analysis where you look at the cost of the individual animal versus the gain for society, EI research and management. And within that, of course, applying the 3 Rs. Together with other researchers, I've proposed that there should be an ethical committee specialized in wildlife, a wide specialization in there. It should be about animal welfare, animal ethics, species biology, population health, and of course public health. Now we talked about management, where it's gaining knowledge. If we widen it a little bit, you can also look at 3 Rs in other situations where you don't necessarily have to gain knowledge. For instance, when you cull for disease control or pest control, we're actually trying to, for instance, fence off badgers so don't get close to cows. When it comes to TB, or when you're trying to repel rodents to enter an area instead of kill them, and then of course use the less invasive the method that causes less stress and suffering when you do kill them. Now, the final words here is, regardless of where you put this dividing line between research and management, and what legislation these animals falls under, there is a need to ensure animal welfare and the use and the development and inspiration to develop these 3 Rs. And in fact, there was an international conference in Norway that in its consensus document, and this was about field research, stated and proposed that animal welfare and the 3 Rs are important regardless if it is about research or management. So with this bear that has all refinement you can think about, Åsa will probably mention it later, including oxygen, I'll finalize my talk and see if there's time for a question.
Thank you, Johan, for a very interesting talk. We have time for 1, maybe 2 questions.
I'm Outi Vainio from the University of Helsinki. So you mentioned that the ethical committee, which would be specialized in wildlife research, would be needed. Would you give a little bit more reasons why we should have it?
Yes, we believe, at least in Sweden, there is a lack of knowledge how you actually try to review research with wild animals, and that's actually stated by the Board of Agriculture itself quite recently. We would like to widen that and say not only that we today classify as research animals should be included. Any kind of activity that is similar to what you put through these animals through should be reviewed and approved ethically. And with that, you would then gain the 3R, the development of the 3Rs, for instance. And the broad expertise that I tried to summarize there, the idea about that is that when you have wild animals, they are, for instance, they are in the wild, they're not in a controlled area. So you don't just need a vet or someone, a ethologist or an animal welfare knowledgeable person there. You also need someone who knows what's happening in nature when you release these animals back, for instance. What's the risk of disease transmission, and what can happen if the diseases transmit to humans, the One Health concept, and how can it actually affect the environment itself. So that's the idea behind that, to have a much broader expertise in that kind of committee that we call it.
Yes, let's take the last one.
Thank you. You mentioned that for endangered animals we should use a strict minimum. How then are we to imagine a difference between a strict minimum and a minimum for less endangered animals?
Ask the legislators of the EU Commission. I haven't thought about that, and I think there is no threshold. It's just stated in the EU directive that you should be very thoughtful and try to minimize specifically these endangered animals. I don't have a better response than that. But maybe, have you thought about it more so you can elaborate on it? No? Because I would like to know myself.
Thank you very much.
Someone else.
Johan.
Okay.
Thank
Thank
you.
you.